Welcome everybody to the 40th edition of the Multiomics Reading Group. I'm your host for today, Cesar Miguel Valdez Cordova. And today, we have the absolute pleasure to present, or rather, not to present, but to host both Ellie Haber and Shahul Alam, who will be presenting Heimdall, their modular framework for tokenization and single-cell foundation models. Sinu will be giving an introduction, as she often does. Sinu, please, if you may.
Yeah, sure. Thanks, Cesar. Hi, everyone. Welcome to our reading group today. So Ellie is a fourth-year PhD student in the machine learning department at CMU, advised by Gemma. And her research focuses on understanding how and why single-cell foundation models learn, with the goal of identifying the inductive biases and also failure modes that can determine when their representations from the foundation model generalize or how to improve them. She's also interested in developing machine learning methods for spatial transcriptomics that can support cross-platform integrations and also enable deeper insights into the cellular organizations and tissue environments. And also, we also have our second speaker today. Shahul is a final year PhD student, also from the CMU, advised by Gemma from the computational biology department. His research focuses on the development of AI algorithm for modeling and interpreting single-cell sequencing data. He is also particularly interested in designing biologically grounded machine learning algorithms that can derive insights about, like, for example, tissue architectures and functions that can be derived from spatial transcriptomics data. He also leveraged tools ranging from non-negative matrix factorizations to transformers to this end. So let's welcome them and thank you for accepting our invitation. And it's all yours.
Yeah, thank you for the intro, and thank you guys for the invitation to present our work. We're both really excited to share this with you. So yeah, my name is Ellie, and today Shahul and I are going to be presenting on our work Heimdall, a modular framework for tokenization and single-cell foundation models. So to start, single-cell foundation models in the past few years have really been advancing rather quickly, but it still remains very challenging to attribute which design choices actually drive performance and generalization, especially when design choices such as tokenization, pre-training data, and training recipes vary simultaneously. Heimdall is our attempt to make that attribution possible. We decompose tokenization into interchangeable modules and run controlled ablations across tasks to turn model comparisons into actionable design insights. In terms of structure for the presentation, I will walk through the core idea behind Heimdall, as well as discuss some key empirical results from the paper. And Shahul will give a walkthrough of the Heimdall code base, showing how to use it to evaluate existing tokenizers and how to implement and test new tokenization modules within the same controlled framework.
Hi, Ellie. Sorry, just to interrupt you. Just to make sure, the slides is rolling. Oh, okay, you are turning to the second page. Sorry for that.
Yeah. Oh, no worries. So to motivate why tokenization matters in single cell, let's start with how foundation models work in natural language. In language models, everything is really built around this core idea that a sentence is a sequence of discrete units, words or subwords. Tokens are mostly identity, and the order carries inherent meaning due to the sequential nature of sentences. So design choices like positional encodings or pre-training objectives such as autoregressive or masked objectives are a natural design choice in these scenarios. But when we move to single-cell biology, this analogy breaks in two ways. First, a cell is not a sequence. It's really a set of genes with no inherent ordering. And each gene isn't just an identity as well, it also comes with a continuous expression value that we have to consider. So if we want to build single cell foundation models, we have to not just make design, we have to essentially be making design choices that aren't necessarily intuitive to the data. This poses some questions such as, how do we represent gene identity? How do we represent expression? And more importantly, how do we impose an order, if any at all, on data that fundamentally is not sequential? And this is why tokenization really ends up being a first-order design choice in single-cell foundation model development. Given these challenges, it's helpful to look at how the major single-cell foundation models that currently exist in the literature actually make these choices in practice. All of them ultimately, even though they all differ in a variety of different ways, they do still have to answer this question of how to convert a cell into a transformer-compatible sequence of tokens. But like I mentioned, they all do so in a variety of different ways that make it hard to compare. So for example, let's start with the single-cell foundation model GeneFormer. GeneFormer treats genes as discrete tokens and learns an embedding for each gene in its vocabulary, very similar to in natural language processing. The key challenge here, though, is ordering. Since a transformer expects a sequence, GeneFormer resolves this by using expression primarily to rank genes. Genes are ordered by their expression within the cell and normalized by the gene's median across all cells. This design choice allows for prioritizing more informative or differentially expressed genes in the sequence, so that the model is able to see an ordered gene list rather than an unordered set. So in GeneFormer, expression is primarily entering the cell representation indirectly, not necessarily as its own token feature, but as the mechanism that determines which genes get prioritized early in the sequence. That's really the core idea behind how GeneFormer resolves this question of cell tokenization. So scGPT also treats genes as tokens, but it makes a different design choice in regards to how it handles gene expression information. Instead of using expression mainly to impose an ordering over genes, scGPT represents expression explicitly by discretizing each gene's expression into bins, and then it embeds these bins. As a result, the input element for each gene is which gene it is and its expression measurement for that gene. This means that expression is available directly as part of the token representation, not only indirectly like in GeneFormer through the sequence order. UCE makes two distinctive tokenization choices that clearly distinguish it from a lot of the design decisions that other single-cell foundation models make for tokenization. First, for the gene identity, unlike how GeneFormer and scGPT handle it by using random embeddings, UCE actually attempts to incorporate a biological prior. So in this case, gene identity is represented via initialization using ESM2 protein language model embeddings. So the gene token is essentially starting with information beyond just a pure learned lookup. Conceptually, this is a plausible advantage in generalization settings, for example, especially cross-species transfer, where conserved protein properties can often provide a shared signal. Second, another way in which UCE differs from the tokenization strategies I've already discussed with GeneFormer and scGPT is that UCE treats, rather than ranking the genes, for example, like GeneFormer or using an explicit expression bin for each gene, UCE uses expression to define a sampling weight. So genes with higher expression are more likely to appear and appear frequently in the model's input sequence. And because the transformer still requires an ordered sequence, UCE imposes order after sampling by sorting genes by genomic position, for example, using chromosomal location. So we can see here that across these three example single-cell foundation models, GeneFormer, scGPT, and UCE, there isn't a single obvious way to tokenize a cell. Models make fundamentally different choices about what information is carried in a token and how the sequence is constructed. But unlike with natural language, these are actually, it's not necessarily intuitive or clear what the right approach is. The challenge, especially the complications with this challenge, is that it's really hard to know what choices are actually beneficial or which aren't adding any useful signal or which are actually detrimental, and in which evaluation regimes that this is true. One way that the single cell community has tried to answer that question is through evaluation studies, which I'll go discuss a bit more in the next slides. So a natural question is, don't we already know which single cell foundation models work best? This is partially true. There are several evaluation studies that have come out in the past few years that have been extremely compelling that compare single-cell foundation models across a wide range of downstream tasks. These tasks include batch correction, cell type annotation, perturbation prediction, network analysis, and so on. And these comparisons, importantly, provide rankings of how these models perform on these tasks in relation to one another. But the challenge is that these models differ in a variety of different design choices, whether that's the gene identity representation, expression encoding, ordering, what data and how much data it's pre-trained on, pre-training objective, the list goes on. So the problem here is that when a model performs well in these evaluation studies, the ranking doesn't necessarily tell us why. We don't know what design choices actually contributed to that gain. So even though we have these very interesting leaderboards now of how these models compare to one another, we don't yet have mechanistic insight. We don't really know the design rules that tell us what changes we should make so that the next single-cell foundation model that comes out can reliably prove to be better than the previous iterations. And there's a very concrete reason why this matters, because in some evaluation scenarios, simple baselines can look surprisingly strong compared to these single-cell foundation models that are pre-trained on millions of transcriptomes and take up, hundreds of GPU hours. So here's an example of where this shows up a bit more clearly. In this evaluation study that came out in 2023, a simple logistic regression baseline is competitive with and sometimes even outperforms single-cell foundation model scBERT across training data sizes in this cell type annotation task. You can see that in both the accuracy and F1 metrics. Results like this show that today's single-cell foundation models consistently outperform do not consistently outperform strong simple baselines in some evaluation regimes. But the takeaway is not necessarily that the foundation model paradigm is misguided. The underlying idea, that large-scale pre-training to learn transferable, generalizable representations for single-cell biology is still very compelling, but the issue is that we don't yet have these reliable design principles for how to realize that promise in single-cell biology. When we are only really reporting these end-to-end rankings, we learn, which models perform better, but not which modeling choices are responsible for the difference. And without this attribution, it's easy to mistake confounded implementation details for genuine progress. And it becomes really hard to systematically build the models that will actually beat these strong baselines, because this is a problem that we've seen for a variety of single-cell foundation models in various studies that continues to come up. So the question really becomes, how do we turn model rankings into these actionable design rules? In our case, Heimdall is the approach that we took to answering this really important question. So by isolating tokenization from the rest of the training stack and testing its components in controlled ablations, we attempt to do just this. So the core idea of Heimdall is to separate tokenization from the rest of the training stack so we can isolate tokenization's effect in particular. Instead of comparing the end-to-end models that differ in multitude of ways at once, we treat tokenization as a set of explicit design choices, implement them as modular components, and then swap them systematically to measure which choices reliably move performance under a controlled evaluation pipeline. In Heimdall, tokenization is factored into 3 parts. First is the gene identity module, F of G. What representation does the model use for a gene? Is it purely learned embeddings, or does it incorporate biological priors, such as, for example, with UCE, the protein-informed representations? Second is the expression encoding module, F of E. Here, the choice is, how do we encode expression values? Are we discretizing expression into bins? Do we encode it as, say, a continuous value embedding? Then finally is the cell sentence constructor, F of C. This decides, how do we turn a cell into actual inputs to the transformer? This includes how gene identity and expression are combined, and critically, how do we impose an order on inherently unordered sets of genes? F of C also covers practical choices like context length and how we reduce token level outputs into a single cell embedding. The goal of Heimdall is to make these choices explicit as well as mix and matchable so we can run controlled ablations, changing one component at a time and identify what actually drives single-cell foundation model performance. Now that we've defined the 3 tokenization components, Heimdall operationalizes them as plug-and-play modules. The key claim of Heimdall is that the major single-cell foundation model tokenizers that exist in the literature can be described as one choice of f of g, one choice of f of e, and one choice of f of c. Heimdall makes these choices explicit in the coding configuration so that they're modular, swappable, and comparable under controlled training and evaluation frameworks. On the left are some examples of existing f of g's, for example, which define what it means to represent a gene. So like I kind of mentioned in previous slides, you kind of have this divide of, random embeddings versus pre-trained embeddings that encode some sort of biological prior. So here are 2 additional examples that exist. For example, scBERT uses gene-to-vec embeddings, which model gene co-expression. And there's also this example of GenePT as well, which uses NCBI gene description data. For f of e, which is how the expression is encoded, this difference between binning or to discretize expression or using continuous embeddings. And then importantly, for f of c, we subdivide this for additional granularity into 3 submodules. So in this case, we have the order submodule, which is pretty intuitive. It's how you assign an ordering to the genes, whether this is expression-based sorting from GeneFormer, random sorting, or even chromosome sorting from UCE. Sequence is how we actually form the input sequence from that ordering. So are we simply reordering the genes up to context length, or do we resample genes with weights so that highly expressed genes appear more frequently, like a kind of like bag of words modeling from NLP? And then reduced is the idea of how we pool f of g and f of e embeddings into a single input into the transformer model, whether that's through identity pooling, summation, etc. And so the key idea here is that once you view tokenizers through this framework, models like GeneFormer, SCGPT, and UCE are no longer these monolithic systems. There are specific configurations across f of g, f of e, and these f of c submodules. And this unlocks 2 things. First, we can build hybrid model tokenizers by recombining components across these models, and we can run targeted ablations by changing one module at a time. For example, taking SCGPT's f of g and combining that with UCE's sequence function or GeneFormer's order function in order to identify which design choices actually drive performance. Once tokenization is expressed as a set of modular choices, the next question is how to evaluate these choices in a way that actually reflects generalization, not just in distribution accuracy. So in order to kind of highlight how Heimdall is useful for evaluating these tokenization choices in single-cell foundation models, we evaluate across a variety of different downstream evaluation tasks. So we evaluate across cross-tissue, gene panel, and cross-species generalization, as well as reverse perturbation prediction in our paper. These settings are really there we design these to expose when tokenization really helps or hurts transfer learning. But in this presentation, we focus on the following evaluations, and we will focus in particular on key results that illustrate design attribution, as well as how Heimdall enables systematic ablations in hybrid tokenizers. So here's our first evaluation setting, cross-tissue generalization for cell type classification. Here we train on cells from one tissue, in this case gastrointestinal, and test on a different tissue, brain, so that the model has to transfer across a substantial distribution shift. Throughout these classification experiments, we report Matthews correlation coefficient, or MCC, because it's more robust than accuracy under class imbalance and multi-class settings. For the tokenizers we compare, we focus on a representative set of widely used canonical single-cell foundation models. So in this case, we're focusing on GeneFormer, SCGPT, UCE, scFoundation, and scBERT. The goal here isn't to be exhaustive because there have been many single cell foundation models that have been coming out over the past couple of years, but rather the goal is to cover the major tokenization patterns that repeatedly show up within the literature. A quick notation point also is that the -tok suffix that you can see on the x-axis of the plot means that we've re-implemented that model's tokenization strategy inside the Heimdall framework. So these are tokenizers evaluated under the same training pipeline, model backbone, and optimization settings. Only the tokenizer across these models is what's changing. What you can see here is that a simple linear baseline using raw gene expression, the red horizontal line, either matches or outperforms the current single cell foundation model tokenizers in this cross-tissue setting. This shows that in this evaluation regime, the tokenizer choices alone are not reliably buying generalization beyond a strong baseline. This is exactly why Heimdall matters, however, because since the stack is controlled, we can now run targeted ablations to change the tokenization design while keeping everything else fixed and identify which components help and which components don't improve performance. Finally, also an important experimental detail, these results are trained from scratch with no pre-training so that we can run many controlled experiments efficiently. In the next slide, however, I'll show a limited pre-training scale ablation to confirm that the directional conclusions that we draw from these tokenizer comparisons still hold when we add small scale pre-training. Here we evaluate whether simply scaling pre-training improves downstream performance for these tokenizers. On the x-axis is the amount of pre-training data that we incorporate, whether that's from 0 cells, which means no pre-training at all, just training from scratch, up to 1 million cells. For each point, we pre-train the model at that scale and then fine-tune on the same task as in the previous slide, the cross-tissue cell type classification, training on the gastrointestinal and evaluating on brain tissue. The main pattern here is that increasing pre-training scale yields only modest improvements in MCC, and crucially, the relative ordering and gaps that we observed in the from scratch settings are essentially preserved. In other words, adding pre-training does not materially change the conclusions we would draw from the tokenizer specific comparisons. The same tokenizers remain stronger or weaker, and the direction of the effects is stable across scales. Methodologically, that's quite useful. It means that we can run many controlled tokenizer ablations in a low or no pre-training regime and still recover the same directional design effects, and then later reserve full pre-training for a smaller set of conformations, which allows us to run a much larger variety of different ablations as well in a more efficiently, essentially. With that validation in place, we can now ask a more mechanistic question of, if scale isn't the main story, what tokenizer design knobs are actually moving the performance? One obvious lever is context length, how many gene tokens the model is allowed to see per cell. So now here we ablate context length while keeping everything else fixed, same backbone, optimization, training setup. The only change here is how many gene tokens the model is allowed to see per cell. A few patterns stand out. For SC-BERT, UCE, and SC Foundation, performance improves as we increase context, but it tends to stagnate around roughly 2048 tokens, while SC-GPT continues to improve with increased context length. Beyond that, though, for the other models, marginal gains were small as we increased beyond 2000. A simple intuition for why this is that many cells have on the order of approximately 1,000 expressed genes due to the high sparsity of scRNA-seq data. Once the model's input is large enough to capture most of that expressed signal, additional tokens increasingly add redundancy and lower signal genes. So we are getting diminishing returns by adding context. GeneFormer behaves differently, however. Its performance is best actually at the shortest context, around 256 tokens, and then its performance degrades as we increase context before plateauing around 1,024 tokens. This pattern suggests GeneFormer's advantage is not coming from seeing more genes, but rather it's consistent with GeneFormer benefiting primarily from which tokens appear early in the sequence due to its expression rank ordering strategy, which is especially valuable when the sequence budget is tight. As we increase the context length, we start including lower priority genes that are adding lower signal, and the ordering advantage is getting diluted. So that's why we believe we're not seeing those same monotonic gains. Next, we'll isolate ordering specifically to quantify how much of gene former's behavior is explained by its expression-based ordering so that we can try to confirm this hypothesis. On this slide, we do a targeted ablation of gene former's order module, holding the rest of the tokenizer and training stack fixed, and ask how much of gene former's performance comes from how the genes are ranked within the input sequence prior to input to the transformer. This is exactly the kind of analysis that Heimdall enables that end-to-end benchmarks typically cannot. Because it isolates a single design choice under a controlled comparison, we can do this attribution of design choices. So in this plot, the bottom gray bar is gene former as its tokenizer as implemented in Heimdall. And then we swap out gene former's default expression-driven ordering for alternatives. For example, UCE's chromosome-based ordering or random ordering. As we can see here, by doing this swap out of gene former's original expression-based ordering for these alternatives, we see a significant performance drop, with the largest drop being under random ordering. This is the key value, really, of Heimdall. In a typical evaluation study, gene former differs from other models in so many different dimensions simultaneously, so you can't attribute the gap in ordering versus something else. Here, because the comparison is controlled, we can directly conclude that ordering is a major contributor to gene former's gains over, for example, other hybrid tokenizers. Mechanistically, gene former's ordering strategy front-loads the sequence with the most informative genes, as we discussed previously, which is especially valuable if you want these like smaller finite context budgets in order to increase compute efficiency. And because this advantage is appearing with shorter context and we're still getting better results, as we saw in the previous slide, like I was mentioning, this could lead to better compute efficiency without sacrificing performance, because in some existing single cell foundation models, for example, SC-BERT, they actually, instead of using like the transformer architecture, they use performers so they could add increased context length. But in this scenario in particular, we're seeing that increased context length isn't actually leading to informative gains. So the next evaluation is reverse perturbation prediction. The goal here is to predict the gene knockout label from a control to perturbed cell pair, and we report MCC. The paired cell task really is forcing the model to represent subtle distributed expression changes induced by genetic interventions. In practice, this is exactly the type of setting where tokenization decisions are really crucial in order to help preserve important perturbation signal. On the right is test MCC on this task across these 5 representative single cell foundation models. And again, as before, the -tok suffix means the tokenizer has been re-implemented in Heimdall and evaluated under all the same framework so that we can attribute these design choices effectively. In this scenario, we can see a clear separation. Tokenizers differ substantially and outperform the linear baseline. So unlike the cross-tissue setting we saw in the prior slides, where tokenization alone didn't reliably beat a strong baseline, this perturbation regime makes it clear that tokenizer strongly impacts performance. Notably here, we can see that UCE performs substantially worse in this perturbation setting, making it a particularly informative case study. The gap here is large enough that we can treat it as, say, a failure mode that we want to diagnose and improve upon. Now, a standard benchmark would stop here and simply, report that UCE is worse. Heimdall, though, lets us go one step further and actually ask why. I think someone raised their hand.
We have a question.
Yes, thank you. I have a question just regarding the test experiment. So is the test data made of just cells for which the KO gene was already in the training data, or is the test set made of cells for which the KO gene was not in the training data?
Sorry, can you repeat that one more time?
Yeah, just so regarding the test set for testing the performances. So we could say that one strategy to split the train and testing data is just to do a random sampling. And both in test set and in training set, we'll have expression of cells that were perturbed for CCR7. But another strategy would be for the training set, we take all the cells that were perturbed for this set of genes, and for the test set, we take all the cells that were perturbed for another set of genes, for which the intersection of the set of genes is empty. So that would be another strategy. So which of these two strategy did you use for testing the performances here?
We used the first strategy that you talked about.
Okay. So it's really being able to reconstruct the expression level for a KO gene rather than being able to predict what would be the expression level for a new gene that was not seen during training.
Well, so the idea behind, and correct me if I'm misinterpreting your question, please. But so what differs, so in a lot of single cell foundation model evaluations for downstream analysis, one of the common tasks is perturbation prediction. So given a particular knockout, you want to reconstruct the new perturbed expression vector from the unperturbed. But in particular for the task of reverse perturbation, which I believe was first introduced in the SC-GPT evaluation paper, and then also used in cell PLM, is the idea that you want to predict the particular gene knockout label as opposed to the actual expression vector. And so in our case for reverse perturbation, unlike what we did in the cross-tissue classification for cell type annotation, where it's given one cell, you want to predict its label, here we used an aggregator to aggregate the expression vectors of a perturbed and an unperturbed cell as input so that we could then predict the particular knockout that was occurring, so that particular label.
Okay, great, thanks. Yeah, indeed, I missed the fact that it's a reverse perturbation. So now your answer makes lots of sense. Thank you.
Okay, good to hear.
Yeah, sorry. I also have a question. Just as mentioning, like I'm wondering, like the data used here is real the perturb-seq data, or it's just single cell data? But because every single cell data, they have the gene expressions, and you just know that gene is in the cell and just virtually knock it out. For example, by masking the gene expression, that's one way to virtually mask it out. Or you really use the perturb-seq data?
So we use it's the Norman data set for perturbation prediction. I think it's like a pretty common single cell knockout data set that's been used in a variety of these papers.
Yeah, what I'm asking is because the perturbation data, they are not paired. So I'm wondering how do you choose the perturb expression here? Or you have a bunch of control cells and bunch of perturbation cell and try to encode them all at once and then somehow aggregate them together?
Yeah, so the idea is, and we have more details on this also in like the supplementary material of our paper, but the idea is that we have a variety of these control cells and perturbed cells, and we essentially create these like matched pairs so that we can make this an actual paired task.
Okay.
Yeah, because we wanted to extend our evaluation to not just work for single cell tasks, but also paired cell tasks as well.
Yeah, thanks.
Yeah. Yeah, so, is there another question? Yeah, go ahead. I think there's a hand.
I have a simple question. Mostly, most of the single cell data sets is really huge. Did you use the full data or a subset of the data set?
We took a subset. So the Norman data set is quite large. So I believe we subset to, I think it was approximately 100 different unique gene knockouts. And then we subsampled, but This was the largest data set we ran on, so these experiments definitely took the longest because we were trying all these, even though so it's 100 knockouts, and then we're trying this in a variety of different cell pairs. I don't remember the exact number of cells we ran on for this, but we definitely subset from the Norman data set because otherwise it would have taken way longer.
Thank you.
Yeah. Right. So I guess just to recap this slide before I go to the next one. So we're noticing here that UCE performs worse in this perturbation setting compared to the other tokenizers. So this makes it an interesting case study for us to evaluate why this is occurring using Heimdall. And so here we run targeted ablations, changing one component of tokenization at a time while holding the rest of the stack fixed so we can pinpoint which design choices are responsible for the drop in MCC. So on the left here, we did an ablation on F of G, gene identity. We can see here that really the main message is that a strong biological prior often helps. Using a protein-informed identity embedding such as ESM2 in the original UCE tokenizer beats a random identity initialization with significance, and we see similar behavior with other informative priors. Using Heimdall, we also are able to uniquely plug in additional pre-trained F of G options. For example, hyena DNA provides a DNA sequence-based prior, and GenePT provides a text-based prior derived from NCBI gene descriptions. The conclusion here is that F of G is a meaningful module in tokenization for this particular task. Next, on the right, we ablate expression encodings F of E. Here, the result is very clear. Adding an explicit expression representation, whether continuous or some form of binning that occurs in these different models, for example, auto binning from SC Foundation, integer binning from SC BERT, or quantile binning from SC GPT, leads to a substantial MCC boost relative to UCE tokenizer's baseline behavior, where expression primarily enters the cell representation indirectly through gene sampling. So for this perturbation task, the model benefits when expression is represented directly, not only used implicitly. Finally, we ablate the F of C order submodule, UCE's default choice, sorting by chromosomal position. And we can see here that this is not optimal in this regime of reverse perturbation prediction. Replacing the chromosome order with either random sorting or expression sorting from GeneFormer improves performance, suggesting that the chromosome ordering is not the right inductive bias in this evaluation setting. Putting these pieces together, the best performing configuration is effectively a hybrid. If you say keep the strong gene identity prior with ESM2, but add explicit expression features and use an alternative ordering rule, you could improve UCE's performance significantly in this paired cell task of reverse perturbation. And this is exactly the kind of design attribution Heimdall is meant to enable, not just answering the question of, UCE versus SCGPT, but which tokenization decisions are responsible for this performance gap and what concrete changes improve the model. Next, we look at cross-species generalization for cell type annotation, in which we train in human and evaluate on mouse data. The first thing to notice is the separation. UCE is substantially higher, while most other tokenizers are near chance level MCC, meaning they transfer very little usable signal across species. So this isn't a small ranking difference. Rather, UCE appears to be operating in a qualitatively different regime where cross-species discriminative structure is preserved. This immediately suggests a concrete hypothesis about what's causing this gap. UCE's most distinctive tokenization choice is its gene identity representation, initializing genes using ESM2 protein language model embeddings, which provides a shared conserved signal across species. In this next slide, we further test this hypothesis by keeping everything else fixed in terms of the tokenization modules and swapping only the gene identity F of G representation of each of the other single cell foundation model tokenizers and switching it to ESM2. On this slide, we run this controlled F of G ablation. The solid bars are the default tokenizers, and the striped bars are the same tokenizers after we replace only the gene identity F of G with ESM2. Everything else in Heimdall stays constant. The pattern here is very consistent. Adding ESM2 gene identity boosts cross-species performance MCC across essentially every tokenizer, even some of them outperforming UCE. Several methods that were previously, near chance performance are moving into like a meaningfully better regime once they share the same protein-informed gene expressions, gene representations. And this supports our hypothesis from the previous slide, which is that a large fraction of the cross-species gap is driven by gene identity priors. And it also gives us a practical takeaway. If cross-species transfer is the goal, gene identity representation is a first-order design lever that can lead to practical conclusions for our hypotheses on tokenization. So to wrap up this part of the presentation, the gap that we started with is that prior single-cell foundation model evaluation studies are good at ranking these end-to-end models, but they don't isolate why one model wins when all of these different design choices vary quite significantly across models. Heimdall is our response to that through factorization of tokenization into explicit swappable choices from F of G, F of E, and then the F of C submodules order, sequence, and reduce. And it allows us to evaluate all these different choices under standardized pipelines so we can run controlled ablations. Across the evaluations we observed, the key finding is that the performance differences are most often driven by a small set of tokenization levers, most consistently the gene identity priors F of G, and how expression is represented either explicitly through F of E or implicitly through ordering. And because we can swap the components one at a time, we can go beyond this idea of, say, model A beats model B, and actually attribute which design choice causes model A to beat model B. A second takeaway is that hybrid tokenizers can outperform single model defaults, as we saw with UCE and reverse perturbation. Mixing the strongest components from different approaches can yield better generalization than copying any one canonical tokenizer end to end. And this is something that Heimdall allows us to observe that otherwise in the literature hasn't really been fully actualized. Looking forward, there are 3 directions that we're pretty excited about. First is broadening the benchmarking across more models beyond the 5 representative models that we chose, as well as different data sets. Second, we hope to scale up to full pre-training and explore alternative pre-training objectives and architectures so that we can also provide more best practices on how to approach pre-training for single-cell foundation models, as well as other model architectures. And third is extending the module library to incorporate more multimodal priors, not just RNA or the ESM2 protein informed embeddings. And finally, something that is pretty exciting about Heimdall is that, for people who are interested in single-cell foundation model development, if you have your own hypothesis about tokenization, say a new gene prior, expression encoding, or new ordering rule, Heimdall is designed so that you can implement that module and test it side by side with existing tokenizers under the same controlled evaluation. So now I'm going to pass it off to Shahul, and he's going to go more in depth on how to actually do this with the Heimdall framework.
Okay, thanks, Ellie. So I'm going to cover this part relatively quickly, so we have some time for questions. But this is basically a demo of how to use Heimdall and the actual code base to accomplish what we presented in the first part of the presentation. So Heimdall uses the Hydra configuration framework to set up each tokenizer and each experiment that we're interested in evaluating. So the first part of the configuration is to define a shared experimental context that we can use to evaluate all of the tokenizers. And the second part of the configs is to define the tokenizer itself. So let's look more closely at one of our experimental configs.
Oh, sorry.
Yeah, so so we'll dive into each of the subconfigs here really quickly. Next slide. So one thing that's very important for us is being able to specify a data set very explicitly, because many of the pre-trained single-cell foundation models use different pre-training corpora to demonstrate their point that they can train a generalizable model. So here we want to see if we switch out those pre-training data sets, what is the actual performance difference between tokenizers. Next, we define the task, which is used for either pre-training or for downstream evaluation. So here we specify the label. So we pre-compute labels such as the cell type, or in the case of reverse perturbation, as we demonstrated, these would be the gene labels. We also specify the splits, which can be predefined or can be random. And we'd specify the linear head, or whatever head that you want to use to predict the label from the embedding from the single cell foundation model. So we can make that head more or less complicated depending on the complexity of our downstream task. And of course, we can use whatever loss function is appropriate. Another important thing that we want to control for is the size of the model. So each of the proposed pre-existing single cell foundation models has a different architecture and size and depth. So here we can carefully control all of that with a single model configuration. Finally, we've moved on to the tokenizer itself. So this is where we actually design the tokenizer that's unique for that model. Here we're trying to implement gene former. So in this case, the gene tokens are actually randomly initialized, and each token corresponds to a unique gene. So that's what we're specifying here. However, you can also use pre-trained embeddings, as we'll show later. And the FME config specifies how to encode the expression. For gene former, we don't use the expression encoding itself. We only use it for ordering the gene tokens, but not for producing an expression encoding for the genes. And for the FFC, here we specify the 3 parts of the FFC. But first we specify the context length, which is very important again, because each of these pre-existing foundation models has a different context length, which confounds the comparison. So once you define that, we define how we build the sequence. In this case, we're saying that we will build an ordered sequence of gene tokens, not a random set. And next, we specify how we order those genes. Here, it's by expression, so highest to lowest. And finally, we specify how we combine the gene identity and expression tokens for each gene. In this case, we just sum their identity and expression representations to produce one token for the gene. And finally, we can just run the experiment. So here we specify which f of g, f of e, and f of c to use, and that's pretty much it. And we easily get what we're interested in as output, such as the UMAPs and also the MCC on the test set. So that's all very easy. Now, in the second example, apart from, like, in contrast to the previous example where we just want to compare against existing single cell foundation models, here we want to try implementing something new and see how it performs. So this is a natural next question to ask. And we can also do that with Heimdall. So in this case, we were asking, if we replace the gene encoding, instead of as using an identity encoding that's randomly initialized, what if we use pre-trained embeddings from a hyena DNA, the DNA sequence model, where we use the gene's DNA sequence as the input for that? So hyena DNA is kind of one of the early models for DNA sequence embedding, early foundation models. And our hypothesis, which we actually tested in the paper, is perhaps hyena DNA can improve the performance of these tokenizers. So how do we actually go about doing this? It's actually quite simple because we actually provide the bare bones for doing that already. So if you go on, we have this FFG implementation, which simply loads the pre-trained weights from disk. So if you pre-compute the embeddings and store them on disk, you can just load them. However, you can imagine much more complicated embeddings, which might involve during data loading, you might have to compute the FFG itself. And all of that can be implemented here in the FFG class. And finally, we just need to add a configuration file for that new FFG embedding, and then we're ready to run the experiment. So all we do is replace the FFG with hyena DNA, and that allows us to do an ablation study, as we showed in the main results, comparing the result of swapping out hyena DNA with the identity embeddings, the randomly initialized embeddings. So hopefully those two demos give you a sense of how easy it is to use Heimdall. And we believe that Heimdall is very important because better tokenizers are key for building better virtual cells. So we want people to use Heimdall to create the next generation of single cell tokenizers, which can perform better, provably better than what people have done before. And finally, because Heimdall is open source and a very portable framework, we hope that people can easily share the findings that they have to quickly advance state-of-the-art in single cell foundation modeling. So please check out our GitHub. If you want to use it, if you have problems, leave an issue. Yeah, that's it. And thank you to our lab mates, especially Nick, who was our co-first author, but is not here right now. Thank you all. Thanks to them for making this possible.
So let's all take a moment to thank our speakers for the absolutely wonderful presentation, very clear presentation. And that opens up the floor for questions. If you guys have any questions, this is the moment to ask them. Senior, please.
Yeah, thanks, Cesar. So I want to ask, so after all these tasks and evaluations, which tokenizer or which combination you think would be the best across all the tasks?
So I can take this one, Shohul, if that's okay.
Yeah.
But so I would say that across the board, we notice that some tokenizers tend to consistently perform quite well, if not necessarily the best, because we've noticed, since this is a diverse array of evaluation tasks, the top performing modules did shift, but there were some tokenization modules that were consistently pretty strong. So what I would say, for example, is that the expression ranking order from GeneFormer tends to consistently give a pretty strong ordering to the genes as input to the transformer. And this is something that we do notice in a lot of existing single cell foundation models beyond the ones that we observed here. It tends to be the go-to for how people use like a somewhat biologically informed ordering function for the tokenizer. In regard to FFG, for example, what we saw with the cross-species analysis is that it gives a good implicit mechanism for allowing for cross-species analysis. And something that, in my opinion at least, is that if you want a single cell foundation model that truly is generalizable and robust, you would want it to have cross-species generalization capabilities. And it's a pretty low cost effort to incorporate the ESM2 embeddings as opposed to just randomly initialized embeddings. We did notice, though, that pretty consistently, for certain tasks relating to gene generalization, gene-to-vec actually, which we didn't go really into in too much depth in the presentation, gene-to-vec adds a very strong biological prior due to its being pre-trained on gene co-expression. And so even though in some evaluations in the paper, we saw that For example, I believe like a randomly initialized embedding did slightly better than gene-to-vec in some scenarios. In other scenarios, gene-to-vec provided such a strong informative prior that it's a good way of incorporating additional information in a relatively cheap manner. I would say, though, that what we did observe is that the chromosome sorting for UCE seems to be an inductive bias for very specific downstream tasks and doesn't generalize as well to other evaluations. And that is something they actually, I believe, mention in the UCE paper in a later iteration in regard to the chromosome sorting. So while it is a very compelling idea of how to incorporate biological priors, it's not necessarily the best way from our findings as to how to incorporate that information.
Thank you.
Yeah.
Well, Bill, you want to go for this one?
Yeah, sure. Interesting that you mentioned the gene former tokenizer as being like the, let's say, robust or one of the lead ranking strategies. So actually, I have the preprint here. So you're splitting the experiments where you varied context length. It's figure 2F in the preprint. I don't know if you could put it up here.
Yeah, let me go back. Sorry, it's going.
No, it's okay. Yeah, it's fine. And I should have asked, but I had my mouth full.
No, you're good. Sorry, this is.
Yeah.
Okay.
Yeah, exactly. So here you're explaining how, interestingly, for gene former, the smaller context length, the stronger is the model in terms of MCC, and vice versa for the other tokenizers. And you just mentioned how gene former tokenizer was like the one that was often one of the leading rank tokenizers. So my question is that, isn't the inductive bias in gene former just actually selecting genes for which we know expression is high in a given cell? For example, we know that when we pre-process single cell data, we might do like highly variable gene processing. So for any given cell, if you are able to find that gene is kind of likely to maybe be a marker gene for that cell or that cell type, right? So gene former would just be a trick you use to prioritize those genes, right? And then all the other genes, you don't care about them. So if you keep the context length small enough, you avoid the noise that comes with adding other non-necessary genes. Whereas with the other tokenizers, you don't have that little trick. So you're just evolving the context length to make sure you're hitting those genes at some point, right?
Yeah, that's a really good point.
And I think it ties in with one of the original issues you were voicing about like treating a gene expression as a sequential modeling task, which is not, but we're forcing it to look like one, and there are tricks that work. But so yeah, kind of interested in your thoughts there about like what if we just had tokenizers that just looked for marker genes, essentially.
So I think that's a really interesting perspective that you bring up, and something that is important to note is that because you're absolutely correct that a lot of these modeling strategies, they'll select for highly variable genes and things like that. And so in our case, we did not do that level of pre-processing in our analysis. But something that I think ties into your question is kind of this question of, with gene former, if you're ranking, genes by, high expression or highly expressed or highly differentially expressed, those are going to appear very early in the sequence. So if you have a smaller sequence, it's just going to cut off the noise. But something that is actually interesting about a lot of these models is that they have this like inherent sparsity handling. So like in gene former for particular, when it's doing its, ranked expression vector, it doesn't consider 0 expressed genes, which is the vast majority of the genes for a particular cell due to the sparsity of the data. Whereas some of the other single cell foundation models like SC-BERT or SC-GPT inherently attempt to mitigate this by having, since both of those models use binning for expression, they have like a 0 bin, or they have some way of handling the 0 expression. And so in particular, what we're observing for gene former here is that when we increase the context length and we start adding in those zeros, it's adding noise that's diluting the signal from the highly expressed genes. And so and something that's kind of interesting about this is that I've seen in several papers for single cell foundation models, people pose this question of, that's very similar in natural language processing, of how do we extend context length efficiently so that we can input more data and more signal. But in particular for single cell, for these single cell foundation models, this kind of ablation that we did kind of leads us to believe that that isn't as necessary, because the actual signal you're gaining from these large gene expression vectors, like you're saying, primarily comes from those like highly expressed or marker genes.
Okay, so I think part of the answer is that some models handle zero expressions in a efficient enough ways that those don't end up introducing serious signal into the attention layer, let's say.
We also have some examples as well, I believe. Yeah, we do have them in the paper where we compare what happens when you include sparse expression versus just including expressed genes. And we found very consistently across evaluations that incorporating the zero expression, so increasing that context length to include additional signal, actually leads to more noisiness in the results. So it's not actually very informative.
Okay, yeah. So, but back to the idea though that even if you had all these the different tokenizers for which you controlled for zero, like sparsity and zero gene expression, right? Would you still expect to see a graph similar to what we're seeing here right now in terms of gene former outperforming for early, like small context lengths? Because the other observation I made is that if you look gene former at 256 context length, or log 2 thereof, it equates more or less to the best performance for the 4096 context length, right?
Right.
So I feel like there might be like a convergence on both ends. So maybe if, let's say, that graph also had like 128, I wonder if gene former would keep going up in the other direction, up, and then if you went past 4096, so 8192, if the trends would also go up in that direction for all the other models, right? Are we just having two tokenization strategies that are like closing a big circle, but in a bit of a complicated way? Whereas gene former just lays out highly expressed genes in your face, and you can use them right away.
So I think additional evaluation to like further extend context length, or even, like going smaller than 256 to 128, like you're saying, is very compelling. And that is something that, I mean, now that you mention it, I would want to look into just for my own sake of mind as well. But because I do think it's an interesting question. I think based on what we have right here, it does seem like, for example, if we did this same experiment for gene former and we didn't include the sparsely expressed genes, and then we would essentially be adding like additional padding tokens to the end of the sequence, which wouldn't really add signal as well. I think that likely we would see similar performance. I can't really say without running the experiment, but I don't think, based on what we've observed here, that at least for gene former, like increasing the context length would lead to performance gains, because I think that's also most likely why gene former in their manuscript explicitly say that to not include sparse expression, because all of these other a lot of these other models, they, like I was mentioning, like provide this specific handling for it. But it seems, and it's kind of like intuitive, like you were saying, like, so does it make sense for like how you tokenize these cells to just consider like the most highly expressed genes or something because they have the most signal? So I think in that sense, like kind of context length is a function of the tokenizer in a sense.
Right.
Yeah.
Okay.
Can I add something?
Yeah.
So I think part of what you're getting at is like, how do you most efficiently use the context length?
Bless you.
Excuse me.
Oh, bless you.
So like one thing that we're saying is that single cell data sets are inherently sparse, so there's a limitation of how much you can get from adding more gene tokens, because at some point we're just adding tokens. So I don't know if you've seen the state preprint from the ARC Institute. one thing they do to improve the performance on perturbation prediction is they actually use not just the cell that they're interested in, but also a set of similar cells. And they use them together to predict the perturbation effect on that cell. So you can think of that as like you're filling up the context length with more gene tokens, but from other cells. So that could be a way to more effectively use the single cell RNA-seq dataset, given that each cell has a lot of noise and we're probably not measuring all the transcripts. And they have a nice figure, I think, like subpanel in their figure 1, which shows like some sort of scaling law of that type. Yeah,
I see. So some sort of pseudo bulking, essentially, but
yeah.
that you're reconstructing for individual cells at the output. Interesting. Yeah. So yeah, you're increasing the number of tokens that you would actually be expressed, and therefore non-noisy, and therefore enabling larger context length that increases performance through scaling. Okay. Interesting. Yeah, that's an interesting point. Okay. Thanks a lot.
Right, great. How are we doing on time? Can you guys take any more questions?
I can take more questions.
All right, very good. So I want to talk about perturbations, right? I really enjoyed the reverse perturbation framing that you guys put in there, but I couldn't help but think that you have this sort of like two-way problem in the sense that one perturbation can give rise to many different cell states, and the other way around is also true. Like one transcriptional state can also arise from multiple perturbations. Control for that? Did you guys control for that?
So we were, sorry, let me think about this.
It's a little bit of a bender, like, you know?
Yeah, well, so in this case, we were just predicting like the knockout label from the perturbed and unperturbed expression aggregated. And so in that sense, we weren't really looking at, like, cell state wasn't really something we were looking at here. It was more so the particular perturbation expression vectors being induced by a given knockout. Although I think that definitely is, that is something to consider, kind of like in the same sense as, for cell type annotation tasks, like which tissue regions these cells come from, right? So like in our case, we did the cross tissue evaluation, but in a lot of cell type classification tasks, sometimes they subset by tissues before randomly sampling for like training or test. So that is something that could be done. I don't believe that's something we considered here though.
I see. Thank you. And the second part of what I wanted to talk about is more of like an open conversation more than a question. There is a very thought-provoking statement, at least for me, in the very same paragraph, or actually it's like somewhere on page 4, but it says, fundamentally, and I quote, scFMs can only become useful and outperform bespoke methods once they learn robust, generalizable representations of cells. And I would like to talk about, is this completely fair? Because I do think that these models are useful for many things, but at the same time, it is also true that linear baselines really pull their weight. And we don't have this expectation from linear baselines, right, up to a point. We just use the linear baseline for whatever prediction task we need, and we use it and we call it a day. And somehow we have this sort of like additional super expectation of single cell foundation model solving everything for us. So I just wanted to talk about, is this a reasonable expectation? And at the same time, well, actually, once we develop that, I want to talk about linear baselines. But first off, is this a reasonable expectation?
So I guess the way that I see it is, so let me pose the question of, why are people interested in developing foundation models for single cell? In large part, this is due to, the explosive performance that we've seen from these in computer vision in natural language. The transition from traditional machine learning methods to these pre-trained, foundation models in NLP really transformed the field. Like I'm a 4th year. When I started my PhD, like no one, like ChatGPT didn't exist. Like education was different, everything has changed. And so it's like, that has really inspired in large part why people are so interested in single cell foundation models and this virtual cell, the idea of we have all of this data that's available to us. Can we utilize it in some way to create this generalizable representation that we can then use for all these different downstream tasks that we're interested in? And, you know, people saw that worked for natural language. So they're like, why can't that work for, you know, the language of the cell, if you will? And the problem, though, is that with these foundation models, they require so much data and so many computational resources that in a way you kind of have to justify, you know, why you want to use all this additional compute and data and modeling resources in order to use these models. And so when, you know, something comes along and it's like you're comparing these like complicated like million parameter models pre-trained on like millions of 50 million transcriptomes, and it can't be a linear baseline, I think it's something where it's not what you would necessarily expect, right? And I find it like surprising and kind of a thought-provoking way, I guess, because it's like your expectation is not the reality, right? What you expected to perform better is being outperformed by something so simple. And so why is that? And so there are a couple of ways that, you know, I think that's really what that question really is in part what inspired Heimdall. This idea that, you know, I was reading all of these single cell foundation model papers and we would all discuss it, you know, Shahul and our other co-authors and I, you know, SCGPT, UCE, SCVERT, all these models. And we would be asking these questions of, okay, well, what's causing one model to outperform the other? Like, but there are so many different confounders that you can't really isolate it. And so each foundation model paper that comes out, you know, they're like, oh, we're better because we add this biological prior to tokenization, or we pre-train on more, or we have more parameters. But it's so hard to isolate what's actually leading to the gains when so many things are changing at once, right? And so the way that I kind of see it, like going back to the quote from the paper that you said, is that if we want to prove that these single foundation models are kind of, you know, worth their weight and like doing worth the cost and the time that we put into them, we should find ways of understanding, you know, what's causing the simple baselines to beat them so we can actually lead to like those performance improvements. So that statement wasn't necessarily to, you know, like shit on the single cell foundation models, if you will. It was more so to kind of like light the fire of, you know, right now these models aren't working as expected. Like, can we drive them to understand why and like push them forward? If that makes sense.
I absolutely love that answer, and I wholeheartedly agree. Thank you for that. So to like drive it home a bit, what evidence would convince you, or let's keep it concrete, right? What evidence would convince you that tokenization choices matter for the harder generative tasks? Beyond contrasting with like the linear baselines, like let's say you have like a very, so like beyond downstream tests, beyond going like a linear baseline was able to beat this model on this particular prediction test, whatever. So let's.
Oh, I see. So what I kind of find compelling about that is, to be clear, I don't necessarily blame the single cell foundation model papers that have come out for doing the evaluations that they've done, because, well, so the problem is that if you want to evaluate these models, you need a concrete evaluation task to test it on, right? And so these more simple tasks like cell type annotation, while they are more simple, they are quite effective to evaluate a wide scale of models on. But the problem is that these tasks that are more easy to conceptualize and actualize are tasks that the linear baselines are very good at. And so the thing is, single cell foundation models in theory should be able to do tasks. Like if we can find tasks to evaluate them on that linear baselines like can't even touch, if you will, then that would probably also show the effectiveness of those single cell foundation models. But I think what's really stopping the community is that these tasks are much harder to both like conceptualize and test. And like, I think for like, you know, those of us in, you know, working in like machine learning for biology, that's one of those big problems where like the more compelling biological questions we might want to answer aren't really easy to evaluate, right? Like if you're trying to find novel perturbations, for example, with your model, you then have to go and do a literature search or combine with a wet lab and have them evaluate these perturbations. And, that takes a lot of time and resources. And so it is kind of this like predicament of single cell foundation model evaluation where like the easiest to evaluate tasks are also the ones that the linear baselines do very well on.
Wonderful answer, very thorough. So even though I have more questions, I will stop now and I will open up a final round for questions for people in the call. Does anybody else have a question? Okay, I will stop the recording there. So thank you, everybody, for coming out to the 40th edition of the Multiomics Reading Group. Thank you guys again, Ellie and Shahul. Hopefully,
thank you so much for having us.
Thank
See
you.
you soon.
Thanks for the discussion.
Thank you.